This is Seth Valenzuela and Dominic Singh, and this is our project on analyzing the density of whitebark pine cones in the Cascades. Whitebark pine are of ecological significance due to their ability to retain snowpack and regulate runoff, and by doing so, reduce soil erosion and decrease the likelihood of spring floods and summer drought. Clark's nutcrackers play a key role in this process because they are the main vector of seed dispersal for whitebark pine. What makes this relationship special is that Clark's nutcrackers will collect as many as 100 seeds at a time and carry them up to 20 miles away, which helps to expand whitebark pine territory, and store them in caches just below the surface of the soil at the perfect depth for germination. Some seeds are cached in barren landscapes, but luckily for whitebark pine, they can establish themselves in harsh environments that other subalpine trees cannot. Consequently, whitebark pine create an environment suitable for colonization by other subalpine trees by acting as nurse trees and regulating water loss from the landscape. However, due to factors such as climate change, attack by mountain pine beetles, and infection by an invasive blister rust fungus, as shown in Figure 2, whitebark pine has become a species of concern and is now proposed for federal listing under the Endangered Species Act. Because of this, it has become important to keep track of aspects of whitebark pine health, including cone density, which may affect both Clark's nutcracker visitation and regeneration of whitebark pine stands. Figure 4 is a map of our sampling areas. Distance sampling data were taken at locations of bird call monitors, indicated by the blue markers, and were used to track Clark's nutcracker activity. Figure 5 is a diagram of our sampling area. Cone counts were performed by walking transect lines of 100 meters from the monitor in either direction and counting visible cones within a 50-meter distance on either side of the transect line at a 90-degree azimuth from the transect to the tree. And our data were collected in 2021 mostly by lead field tech Emily Hammett, as well as several other volunteers.
For the analysis of this data, we used the software Distance 7.4 Release 1. This software allows for the analysis of different distance sampling models that estimate the density of objects or organisms from data collected through line transect methods. This is done through calculating a detection function equation that is based on the number of observed objects recorded in the dataset in order to estimate the proportion of objects not seen while sampling. Figure 6 shows an example of a detection function for a line transect survey, with the probability of detection being the highest at closer distances and then decreasing over longer distances from the transect line until eventually reaching a point of what is effectively zero detection. This allows for an estimation of how many objects might have been missed in a particular area, which can then be used to better reflect the density of the sampled object in the surveyed area. The parameters of the detection function equation may be defined by 4 key functions: half normal, uniform, hazard rate, and negative exponential, which are then further defined by the series expansions of cosine simple polynomial and Hermite polynomial. One advantage of using distance software in this analysis was its ability to compare the efficacies of object density estimations between models created by each combination of detection function parameters. Another useful feature for this survey was that distance software allows the researcher to specify whether or not the objects surveyed occurred in clusters, automatically taking cluster size averages into account when analyzing cluster data, like those taken in the whitebark pine line transects. We used this parameter option in this analysis as it was the most consistent with the methods used in the whitebark pine surveys and allowed for a greater estimation of density of cones that were surveyed in clusters. Moving to the results, as part of the analysis provided by distance software, the ACIC information criterion values, or AIC values, were calculated for each model. This value is used to compare multiple regression models under different parameters in order to determine which fits the dataset best, with the lowest AIC value indicating the best fitting model. Using AIC value, we found that the best parameters for the whitebark pine cone count dataset were the combination of hazard rate key function and Hermite polynomial estimator, which we show in Figure 7 along with the corresponding histograms for the best estimator combinations found for each key function. Moving to the discussion section, the analyses performed showed great promise for quantifying the validity of cone density estimations and performing secondary analyses to compare cone densities over space and time. Over a longer time frame than allotted for this project, there's potential for future projects to conduct absolute density counts via thorough counts of individual trees that could then be compared with calculated densities. This would take considerable time, but it would provide insight into how accurate the density estimations calculated through distance are. Another possibility is to analyze the density of cones across the surveyed areas of the North, South, and Central Cascade regions. This would indicate any variation in the cone densities found across regions, and may be conducted over many years to determine trends in whitebark pine population and reproductive success. Another possible investigation is to perform distance samples at several shorter transect lengths, such as starting at 50 meters, then 100 meters, and so on, instead of just the 200-meter increments, and comparing the confidence intervals between the surveys. This may be useful in determining how short of a transect length may be surveyed and still provide reasonable estimates in order to maximize efficiency while surveying. And finally, another possible project could be in analyzing the datasets conducted through point transect surveys and comparing with the accuracy of distance to those with line transect surveys. This would be useful in comparing the efficacy of these methods, since some of the tree survey datasets in this project include data collected through point transect sampling. This would also allow for comparisons of cone densities across other tree types that have been surveyed through point transects, such as the Ponderosa pine, to the cone densities of the whitebark pine surveys.
This concludes our presentation on analyzing the density of whitebark pine cones in the Cascades. We would like to thank Emily Hammett and Teresa Lorenz for collecting the data used in the study, as well as U.S. Forest Service for providing the Forest Health Protection Grant. Also, thank you to the Central Office of Student Research for providing the Summer Research Grant.